Hi, my dear student. Welcome to my channel. This session discussing the topic Classical Genetics Part 2 Mendelism. The contribution of Mendel to genetics is called Mendelism. It includes all concepts brought about by Mendel through his original research on plant hybridization. Mendelian genetic concepts are basic to modern genetics. Therefore, Mendel is called father of genetics. Next one, the father of genetics, Krieger Johann Mendel, 1822 to 1884. The best genetist Krieger Johann Mendel unraveled the mystery of heredity. He was born on 22nd July 1822 in Heinz center of Silesia. Now it is called Heinz, Czechoslovakia, that is Austria. After school education, later he studied botany, physics and mathematics at the University of Vienna. He then entered a monastery of St. Thomas at Bern in Austria and continued his interest in plant hybridization. In 1849, Mendel had a temporary position in a school as a teacher and he performed a series of elegant experiments with pea plants in garden. In 1856, he started his historic studies on pea plants. 1856 to 1863 was the period of Mendel hybridization experiments on pea plants. Mendel discovered the principle of heredity by studying the inheritance of 7 pair of contrasting traits of pea plant in his garden. Mendel crossed and catalogued 24,034 plants through many generations. His paper entitled Experiments on Plant Hybrid was presented and published in the Proceedings of the Bern Society of Natural History in 1866. Mendel was the best systematic researcher in the field of genetics. In 1900, the principles of genetics worked by Mendel were rediscovered by 3 botanists, namely, number 1, Hugo de Vries, Holland, working on Enothera. Second one, Karl Correns, Germany, working on Xenia, peas and maize. Third one, Erich von Teschmark, Austria, working on various flowering plants. Mendel's original paper was republished in the Flora, 364 in the year 1901. Patterson confirmed Mendel's work by a series of hybridization experiments. A reason for Mendel's success: he applied mathematics and statistical method to biology. He followed scientific methods. His experiments were carefully planned. The pairs of contrasting character which are controlled by factor. The flowers of pea plant are normally self-fertilized. The hybrids of cotton pea are perfectly fertile. Cross-pollination is not very difficult in pea plants. Artificial fertilization is almost always successful. The genes for the 7 pair of characters are located on 7 separate homologous pair of chromosomes. Many pure breeding varieties are available for the experiments. It is very easy to cultivate the pea plants in open ground. They have a short growth period and short life cycles. He studied the inheritance of only one character at a time. Another one, Mendel experimental system, the cotton pea. Cotton pea is an annual plant. Self-fertilization occurred under normal condition in cotton pea plants. The flowers are large, hence emasculation and pollination are very easy for hybridization. Fourth one, variation. Next one, reproduction, controlled mating, short life cycle, large number of offspring, convenience in handling. So, Mendel selecting the pea plants. Mendel experiments on pea plant. Mendel's theory of inheritance, known as the particulate theory, established the existence of minute particle or hereditary units or factor, which are now called genes. He performed artificial pollination or cross pollination experiments with several true breeding lines of pea plants. A true breeding lines, that is called pure breeding strains, means it has undergone continuous self-pollination having stable trait inheritance from parent to offspring. Mating within pure breeding lines produce offspring having specific parental traits that are constant in inheritance and expression for many generations. Pure line breed refers to homozygosity only. Fusion of male and female gametes produced by the same individual, that is pollen and egg, derived from the same plant, is known as self-fertilization. Self-pollination takes place in Mendel peas. The experimenter can remove the anther, that is called emasculation, before fertilization and transfer the pollen from another variety of pea to the stigma of flower where the anthers are removed. This results in cross-pollination, which leads to the creation of hybrid varieties with different traits. Mendel worked on the study of the pattern of inheritance, and the principles or law formulated now constitute the Mendelian genetics. 7 characters of Pisum sativum studied by Mendel. 4 type of characters: seed, flower, pod, stem. Seed dominant character round, recessive ringled. Cotyledon dominant character yellow, recessive character green. Then flower dominant character white, recessive character violet. Pod dominant character is a full, that is inflated. It's recessive character constricted. Dominant character green and recessive character yellow. Stem dominant character axial pod, recessive character terminal pod. Dominant character tall, recessive character short. 7 characters of Pisum sativum with genes. Relationship between modern genetic terminology and character pair used by Mendel. Character pair used by Mendel, another one alleles in modern terminology, another one located in chromosome. Seed color yellow green. Alleles in modern terminology, capital I and small I, located in chromosome 1. Seed coat and flowers colored white. Alleles in modern terminology, capital A, small a, located in chromosome 1. Mature pod, smooth expanded, wrinkled, indented. Alleles in the modern terminology, capital V, small v, located in chromosome 4. Inflorescence from leaf axil, umbellate in top of plant. Alleles in the modern terminology, capital F, small a, another one small f, small a, located in chromosome 4. Plant height up to 1 meter, around 0.5 meter. Alleles in the modern terminology, L, capital L, small e, another one small l, small e, located in chromosome 4. Undried pod, green, yellow. Alleles in the modern terminology, capital G, small p, then small g, small p, located in chromosome 5. Mature seed, smooth and wrinkled. Alleles in modern terminology, capital R, small r, located in chromosome 7. Mendel 7 characters in cotton peas shown on the plant's 4 chromosome. In number and chromosome, flower color and seed color. Second chromosome, no character. Third chromosome, no character. Fourth chromosome, pod shape, plant height, flower position. Sixth chromosome, pod color 1. Another one chromosome, no character. Last chromosome, seed shape. So the 7 pair of contrasting character locating in the character spray only by 4 chromosome. Mendel worked at the rules of inheritance and arrived at the correct mechanism before any knowledge of cellular mechanism. DNA, genes, chromosome became available. Mendel's insight and meticulous work in the mechanism of inheritance played an important role, which led to the development of improved crop varieties and a revolution in crop hybridization. Can you identify the Mendel gene for regulating white color in peas? Let us find the molecular answer to understand the gene function. Now the genetic mystery of Mendel white flower is solved. It is quite fascinating to trace the Mendel genes. In 2010, the gene responsible for regulating flower color in peas were identified by an international team of researchers. It was called a P gene capital A, which encoded a protein that functions as a transcription factor, which is responsible for the production of anthocyanin pigment. So the flowers are purple. Pea plants with white flower do not have anthocyanin, even though they have the genes that encodes the enzyme involved in anthocyanin synthesis. Researchers delivered normal copies of gene capital A into the cells of the petals of white flower by the gene gun method. When the gene capital A entered in a small percentage of cells of white flower, it is expressed in those particular cells, accumulate anthocyanin pigment and become purple. In white flowers or gene A, sequence showed a single nucleotide change that makes a transcription factor inactive. So the mutant gene form A do not accumulate anthocyanin, hence they are white. Terminology related to Mendelism. Alleles, traits are expressed in different way due to the fact that a gene can exist in alternate form for the same trait. Next one, homozygous. If an individual has two identical alleles of a gene. Heterozygous, that is capital T small t, an individual with two different alleles. Another one, hybrids. Mendel non-true breeding plants are heterozygous. Homozygous recessive small t. When both alleles are recessive, the individual in dwarf pea plants. Homozygous dominant capital T, an individual with two dominant alleles in tall pea plants. Heterozygous tall capital T small t, one dominant allele and one recessive allele capital T small t denotes non-true breeding tall pea plants. Mendelian inheritance. Mendel's laws of heredity. Mendel proposed two rules based on his observation on monohybrid cross. Today these rules are called laws of inheritance. The first law is the law of dominance. Second law is the law of segregation. These scientific laws play an important role in the history of evolution. First law, law of dominance. One factor in a pair may mask or prevent the expression of the other. In crossing between pure homozygous organisms for contrasting characters of a pair, only one character of the pair appears in the first filial generation. Second law, law of segregation or law of purity of gametes. During gamete formation, the genes of a particular character separate and enter different gametes. The hybrids or heterozygote of F1 generation have two contrasting character or allelomorphs of dominant and recessive nature. These alleles though remain together for a long time but do not contaminate or mix with each other and separate or segregate at the time of gametogenesis, so that each gamete receives only one allele of a character, either dominant or recessive. So the second law mentioning that gametes are never hybrid. Third law, law of independent assortment. The genes for each pair of characters separate independently from those of other characters during gamete formation. Evaluation. Question number 1. In his classic experiment on pea plants, Mendel did not use option A, flowering position, option B, seed color, option C, pod length, option D, seed shape. So the correct answer option C, pod length. Second one, the genes controlling the 7 pea characters studied by Mendel are known to be located on how many different chromosomes? Option A 7, option B 6, option C 5, option D 4. So the correct answer Option D 4. Third question, which of the following explain how progeny can possess the combination of traits that none of the parent possess? Option A, law of segregation. Option B, chromosome theory. Option C, law of independent assortment. Option D, polygenic inheritance. So the correct answer, option C, law of independent assortment. Fourth question, gametes are never hybrid. This is a statement of option A, law of dominance. Option B, law of independent assortment. Option C, law of segregation. Option D, law of random fertilization. So the correct answer, option C, law of segregation. Fifth question, select the period for Mendel hybridization experiment. Option A, 1856 to 1863. Option B, 1850 to 1870. Option C, 1857 to 1869. Option D, 1870 to 1877. So the correct answer, option A, 1856 to 1863. Sixth question, among the following characters, which one was not considered by Mendel in his experimentation P? Option A, stem, tall or dwarf. Option B, trichodermal, glandular or non-glandular. Option C, seed, green or yellow. Option D, pod, inflated or constricted. So the answer, option B, trichodermal, glandular or non-glandular. Seventh question, name the 7 contrasting traits of Mendel. Eighth question, what is meant by the true breeding or pure breeding lines or strains? A true breeding line means it has undergone continuous self-pollination, having stable trait inheritance from parent to offspring. Give the name of the scientist who rediscovered Mendelism. Number 1, Hugo de Vries, Holland, working on Enothera. Number 2, Karl Correns, Germany, working on Xenia, peas and maize. Number 3, Erich von Teschmark, Austria, working on various flowering plants. 10th question, what are the reasons for Mendel's success in his breeding experiments?